Hello everyone, this is the fourth lecture on carbohydrates, and today we will talk about different biochemical tests to detect carbohydrates in different samples such as food sample, urine or blood samples, and we will use Fehling's, Benedict and Tollens test. So this test basically tests for the reducing sugar, presence of reducing sugars. So let's first talk about what are reducing sugar. So we have already discussed that in aqueous solution, sugars exist between their equilibrium of their linear and cyclic form. So here, through the process of rotation, cyclic form of sugar will be converted to open form. So in open form, they have free aldehyde group, and this aldehyde group act as reducing agent, and it reduce some compound and get oxidized. So here if you look at this aldehyde group is oxidized to carboxylic group, and because the solution is alkaline here, it will donate its hydrogen and it will become carboxylate ion. So here, since we are taking the glucose, this glucose has now been converted to D-gluconate, and here this copper is getting reduced. So this cupric form of copper has now converted to cuprous form, means 2 plus. It has here the oxidation state of 2 plus, and it's now has become plus. So this is the basic chemistry behind using all these tests. So first one is Fehling test. This is qualitative test. It means we are just saying whether in this sample sugar is present or not. We are not determining the exact concentration of the sugar, and we identify by using this Fehling test. We identify aldehyde groups or reducing sugar. Why reducing sugar? Because they have the aldehyde group. So there are two solutions, Fehling solution A and Fehling solution B. Fehling solution A is composed of copper sulfate pentahydrate. It provides the copper ion. And second one is sodium potassium tartarate, which is made in alkali sodium hydroxide. This sodium potassium tartarate is also called as Russell salt. So here is the basic reaction. This is the sugar molecule with its aldehyde group. This is getting oxidized to carboxyl group. Here basically it is carboxylate ion, and now this copper is reduced and formed the copper oxide. Here its oxidation state had changed. Here this Cu2 plus gained 1 electron and becomes Cu plus. So here oxidation state of copper is plus 1. So if you look here, this is the solution A, means this is the solution of copper sulfate. Its color is blue because of presence of copper ion. Second solution, this is sodium potassium tartarate in NaOH, means this is the alkaline solution. Before the experiment, we mix this solution A and B, and then we add sugar samples to this solution, and then we heat it. After heating, if this changes the color from blue to brick red, it suggests that reducing sugar is present in that solution. If it does not change its color, means after heating still it remains the blue, it means there is no reducing sugar present. So this was the basic concept. Why we should not mix these two solutions earlier? We only mix them when we are doing the experiment, because if we mix them earlier, this copper ion in presence of this alkali NaOH, it will be converted to copper hydroxide and it will form the precipitate. That's why we always mix them freshly whenever we have to do the experiment. So this was all about the Fehling test. So question is, does fructose behave as reducing sugar? Means it does fructose will give positive to this Fehling test? So answer is yes. how the process through the process of tautomerization. So since fructose has this ketone group, and we ketone here does not act as reducing agent, but this fructose is alpha hydroxyketone, means when we have this carbonyl carbon and after next carbon to this carbonyl carbon, if it contain this hydroxyl group, it is called alpha hydroxyketone. And because of presence of this hydroxyl ion in alkaline solution, this undergo the process of tautomerization and converted to intermediate enediol, where its shift is double bond and hydrogen atom. If you look here, this double bond is shifted here and this hydrogen is shifted here. So now what is enediol? When these two carbon are double bonded and these two carbon are attached with this hydroxyl group, they are called enediol. And now it will get converted to glucose and mannose. If you look here, both of these glucose and mannose have this aldehyde group. So now this aldehyde can act as a reducing sugar in alkaline solution. So basically fructose is directly not giving positive to this test, positive result to this test, but it's actually undergoing the process of tautomerization and getting converted to glucose and mannose. And since they have this aldehyde group which is acting as reducing agent, this we can detect fructose through this test. Next one is Benedict test. This is semi-quantitative test. So this is the Benedict reagent. It is the solution of copper sulfate which provide copper ion. Second one is sodium carbonate which make this solution alkaline, and third one is sodium citrate. It prevent the precipitation of copper ion. So this is the same reaction is same as we talked in the Fehling test. So this is the aldehyde group of sugar. It will convert it to this carboxyl group, means it's getting oxidized and it's reducing this copper ion cupric ion to cuprous ion by forming the copper oxide. It is how it is different from Fehling test is, in Fehling test if reducing sugar was present, we got the brick red color, but in Benedict test we get different color on the basis of what concentration of sugar is present. If there is no reducing sugar present, we will get this blue color. If trace amount of sugar is present, we get green color. If we have moderate amount of sugar in our sample, we will get yellow color. If we have high amount of sugar, we will get orange color. And if we have this very high amount of sugar, we will get brick red precipitate. So here we are saying that what amount means, whether it's large amount, high amount, or moderate amount, or very small amount. That's why it is called as semi-quantitative. Still it's not quantitative because we are not determining the exact concentration. So this was all about Benedict test. Next one is Tollens test. This is also the qualitative test, and it is also used for aldehyde and reducing sugar, and also for alpha hydroxyketones, as we already looked in case of fructose. So here we use the Tollens reagent. What is it? It is ammoniated silver nitrate. How is it formed? When silver nitrate react with ammonium hydroxide, it forms the ammoniated silver nitrate. And how it reacts? So it provides the silver ion here, and this silver ion is reduced in presence of this reducing sugar. It will be converted to metallic silver. So here it has +1 oxidation state here. Now it has 0 oxidation state. So when we are adding reducing sugar here with this ammoniated silver nitrate, it will form this silver color precipitate on the wall of this tube, and since it look like mirror, it is called as silver mirror test. So this is the summary for all these three tests. So here in case of Fehling and Benedict, we are using copper as oxidizing agent, and in tollens we are using silver ion as oxidizing agent, and they are reduced to, in Fehling and Benedict, this copper ion is reduced to copper oxide, and here it is reduced to metallic silver. And alkali used in Fehling test is sodium hydroxide, here in Benedict used is sodium carbonate, and in Tollens reagent we used ammonium hydroxide. We detect aldehydes through all these tests, and we also detect ketone only if they are in alpha hydroxy form. And last one is about stability. So Fehling is less stable because we already talked that when you mix the Fehling solution A and B, it will form the precipitate. So this is less stable. Benedict is more stable, and it is commonly used in labs, and also it is semi-quantitative. And Tollens reagent should always be prepared freshly whenever you need to use it. So question is then how to test the non-reducing sugars, like for example sucrose. Since sucrose has this, both of the anomeric carbon of glucose part and fructose part are engaged in bonding, they cannot open up. So for that we must need to do acid or hydrolysis with acid or enzymes, and then we will get this fructose and glucose, and now we can test this. Now it has become the reducing sugar, and we can use all these three tests, Fehling, Benedict or Tollens, to test this non-reducing sugar. That was all for today. Thanks for watching. In next lecture we will talk about the polysaccharides.